Okay, so we've talked about the innate immunity, the non-specific immunity, the immunity that just attacks anything that gets into your body, whether it's a splinter or a piece of bacteria or a virus or anything that gets in. That's innate. You're born with that, non-specific. Now we're going on to adaptive. So the third line of defense is specific, and we call it adaptive immunity. You learn this immunity. This is actually a picture. You have a cell and you have little antibodies, and this is kind of the key of adaptive immunity, is it really revolves around these things called antibodies. Right? So the third line of defense, and again it's specific. So here you have a cell that's making antibodies. Those antibodies attack the invader and help you to kill the invader. There are some main terms you really have to understand or keep in mind as you're going through this. So the first is the term antigen. An antigen is something that's considered foreign to your body or considered not belonging in your body. So your body treats it as if it doesn't belong. It's an antigen. Next are the lymphocytes, and those are the main cells. So lymphocytes are the key cells of the third line of defense, or the adaptive immunity. We talked about macrophages and neutrophils, eosinophils, basophils, blah, blah, blah. Those are all general and vague. They attack anything that gets in. These lymphocytes are very specific. They have to know what they're attacking before they'll attack it. So they could see an invader, but if they're not familiar with that invader, the invader can just walk right on by, cruise right by them. They have no idea. Antibodies are the little tools that the third line of defense makes to fight the invader. We'll talk about the type of cell that makes antibodies and the other types of cells that use the antibodies too. And then memory. The third line of defense, or the real immunity, the adaptive immunity, it revolves around memory. It learns. It gets better. That's why we vaccinate as a child. You give a vaccine, you expose this antigen from a bad, dangerous invader, and the immune system looks at it, recognizes it, figures out what to do with it, and then it knows for the rest of your life what to do in case that thing comes back up. So here you can see kind of a pathway we're going to talk about. We'll talk about the antigens. We'll talk about the different types of cells like B cells and T cells. They're over here. We'll talk about those antibodies and how they work. We'll talk about memory cells, how they stick around. We'll talk about natural killer cells. Then we'll talk about things like antigen presenting cells. So we'll talk about the macrophage again a little bit more. Even though that was in the innate immunity, we're bringing it back just for a short time to show you how the two systems relate. Neither of them stand alone. They depend on each other, but they do different things. So the next terms you need to be able to distinguish between are natural versus acquired immunity. And hopefully the names kind of tell you. Natural means you're born with it. Natural means that when you're born, you have this innate system that automatically attacks everything. Adaptive means you've learned from it. You've learned from your environment. You adapt to the exposed things, or the things you've been exposed to. So if you're exposed to chickenpox when you're a kid, your immune system learns what chickenpox is, and it learns to fight it later in life. So what's interesting is I was watching a lecture not long ago where someone asked an interesting question that said, well, can we basically immuno fingerprint you and tell you where you've been and have this unique characteristic about you? And technically you could. You could kind of tell the background. You could follow, the trace the infections they've had, seeing where they've been. So if there are certain special infections that are unique to certain parts of the world, maybe you could identify what parts of the world they've been in. So it's kind of interesting. We'll probably never use that because we have a much faster, more simpler form of genetic identification by doing DNA samples. So it's a lot quicker. But it's interesting because you can kind of see where people have been or what kind of diseases they've been exposed to by looking at their antibodies that they have in their body, which are identification markers of past exposure to diseases. So we test for antibodies by actually taking a little blood sample. So we can take the blood sample, and then we can look at the types of antibodies they have in their system and know what they're immune to, know the different things that they can tolerate. So then you should be able to describe the difference between active and passive immunity. If you know the term active, it means your body is actually putting in something. It's putting energy into it. Passive means, what the heck? It just allows a fair kind of attitude towards whatever it is. So active immunity means that your body is actually making antibodies. It's exposed to a chemical. It makes an immune system that fights it. Passive means we just gave you the antibodies or the lymphocytes. We just give you the factors. Your body didn't have to work to make those. You just sat on your butt and we gave them to you. So when you get vaccinated, we give you a little sample of whatever that invader is. So now they're giving chickenpox vaccinations, so they shred up the chickenpox. They give you exposure to what the chickenpox looks like, but it's not infectious. And then your body says, hey, this is what chickenpox looks like. Let's make this army against chickenpox. That's an active process your body's doing. Passive, something like rabies gets into you, we give you, because rabies travels fast and it's very dangerous and very lethal. Within 6 days, it can become life-threatening. So we want to make sure that we stop this from spreading. We don't want to give your immune system time to adapt to it. We want to just inject those antibodies directly into you. So we'll just give you a big injection of antibodies that catches the rabies. So that would be passive. We just give you the antibodies, right? So here you can see kind of a breakdown. Here, when you're looking at immunity, you have the innate immunity we've talked about over the last several videos. You're born with it. It's non-specific. And then there's the adaptive or acquired immunity is another word for adaptive. You learn. So you've had exposure to something, and then that's the active. You make your own antibodies, right? Or we can give you the antibodies that are specific for that thing. So one form of passive is that when you're born and you breastfeed, your mom actually gives you antibodies that go straight into you. They're natural forms of antibodies. So that's from mom. We could also make an artificial one. We could take a mouse. We could give a mouse that virus or bacteria, and it can make antibodies. We extract the antibodies and give them to you. Right? So that's another form. So active, passive, innate, adaptive, those are all terms you need to get familiar with. Going back to a concept you already know, but you need to remember where all the lymph cells are produced and developed. So we talked about where all the blood cells are made, and they're made in the bone marrow. It doesn't matter if it's a white blood cell, red blood cell, a lymphocyte, they're all made in the bone marrow. What you need to remember is that they're made there, but they actually don't necessarily mature there. So the two primary lymphocytes we're going to talk about are T cells and B cells. They're made here in the bone marrow from stem cells, hematopoietic stem cells. They turn into the lymphocytes, so they can turn into a B cell or they can turn into a T cell. Those B and T cells get put into the blood, but they don't work. They're traveling through the blood, but they don't work for anything. Once the T cell gets to the thymus is kind of like boot camp. It's training camp for the T cells. It tells the T cell what to do, how to treat the rest of your body. And now we call this an immunocompetent cell. So it can do something. And that T cell goes back out into the bloodstream or the lymph nodes and waits for an invader, a bacteria, something bad to come along. Right? That's a T cell. Matures in the thymus. Made in the bone marrow, matures in the thymus. The B cell is made in the bone marrow. It just moves over to a different area of the bone marrow and it matures in the bone marrow. So they call it the B cell. Right? When we talk about these two different types of cells, we'll talk about cell-mediated and humoral immunities, and it revolves around T and B. Here's just another pathway. You can see it made in the bone marrow, going into the blood. You can see the T cells go to the thymus, get activated, and come back to the blood and go wherever they need to, like a lymph node. Right? You can see the B cells. They're made in the bone marrow. They mature in the bone marrow. They get put in the blood, and they can go to a lymph node. They can go to the spleen. So these things can go hang out and basically wait to ambush bacteria or invaders in these systems. When you talk about the different types of lymph tissue, you've got primary lymphoid tissue, which is the bone marrow and the thymus. That's where the blood cells are made and mature. Secondary is where they hang out once they're matured. So lymph tissue like lymph nodes, the tonsils, MALT, so mucosa-associated lymphatic tissue you see in the GI tract. So those are places the grown-up mature cells will hang out. Those are secondary. So homework number 6, putting it all together. I kind of hinted and slipped a little bit. I knew this question was coming, but rabies is an infection that affects any warm-blooded animal. Rabies' name actually means madness. So what it does is first it has to breach the first line of defense. So how does it do that? You get bitten, right? You get bitten by some animal that's rabid. The rabies hangs out in their saliva and it goes into your bloodstream. So it actually goes into your tissue. It goes into the tissue, and then that's the second question is, so what is the infective agent? Is it a bacteria, a DNA virus, an RNA virus, etc.? How does it get into you? Next is, how does it travel? It wants to go to the brain. What's interesting about rabies is that it's going up to the brain and it's heading for the amygdala, the center that controls aggressive behaviors. So it's going up to the amygdala in the brain, but it's also in your mucus. So you've got this stuff, which I can't tell you what it is. It's the rabies. It's in your mucus. Another part of the rabies goes into the brain and affects the amygdala and makes you more aggressive, so that you want to bite things and spread the disease. Rabies is actually a really interesting disease, the way that it's almost kind of a smart disease. It knows how to spread itself. So you're going to find out that information, and then the last part is how do we treat someone infected with rabies? And like I said, I slipped earlier and kind of talked about this. So when we treat them, how do we treat the infected person? And next is that treatment, is it considered innate, adaptive, active, or passive immunity? So describe it. You can go ahead and pause and come back when you're done with that. All right, so again, know the key players. We've talked about innate, we're talking about adaptive now, and you can see a couple of these kind of overlap a little, and we'll talk about that too. But adaptive immunity, you can see over here the B and the T cells, and we're going to talk extensively about those. We'll talk a little bit about the natural killer cells too. All right, so how does your immune system recognize invaders? This is kind of cool. And this goes back to the antigen-antibody process. All right, remember, we're talking adaptive though. So the adaptive, you've had to have some kind of exposure or have some kind of a knowledge of what combination of fingerprint this invader has. So first you need to define antigen and differentiate between the different types of antigens. So an antigen is anything that's considered non-self. So it's kind of a generic term. It's something your body considers to be foreign or not belonging in your body. And it can be on the surface of anything that doesn't belong. It can even be on the surface of another human being's tissue. So when you get a transplant, you can have an antigen from that other person that comes to you. And your body's not going to like it, so it's going to try and attack it and kill it. So that would be a rejection. So transplant rejection. All right. So there are the different types of infection agents. You can have non-infection. So even dust, peanut. So peanut products that stimulate allergies, those are considered antigens. Bee pollen, different types of drugs, like some people are allergic to penicillin because penicillin has antigens on it and their body recognizes it as not belonging and they attack it. So here's just an example. Let's say that these are different bacteria shapes. Here you have the antigen. So there's the bacteria. On the surface, this is actually called an epitope region. So this antigen's right there. There's another antigen, another antigen. There's one, there's one. You can see they're different shapes. They're kind of unique combinations. When your body recognizes this, it will make an antibody. And antibodies are usually drawn like a letter Y, like this. But that antibody is specific to one antigen, only one. So this one will fit to that antigen, but not to this other two. This one fits that one, but not these other two. This fits that, but not these other two. How about this one, though? What will this fit on? This will actually fit that, the square. It'll fit the round one, and not the triangle one. So these little surface markers here, these are the antigens. Right? To get a good antigen, there are a couple characteristics. First, a large antigen is best. So the bigger it is, the better it is. And it needs to be clearly recognized as foreign, because if it looks like you, it can hide. So if this is a virus that has little characteristics that look like human, your body's antibodies should ignore it. It needs to be complex. So the more different types on it, the easier it is to attack, and it should be plentiful. So hopefully there are hundreds of these so that your body easily recognizes that it's there. If you have only one, it's like a needle in a haystack kind of idea. So I put these little examples here. If I just showed you this, if you were an antibody trying to attack an antigen that looks like this, is kind of a vague thing. You probably wouldn't know what it is. It's kind of a bump. You can see it has a little bit of a shape to it, so maybe you're kind of recognizing it. This is a bad antigen. A better antigen would be an actual three-dimensional cutout windows, wheels that spin, hubcaps, and all that. Lots of details. Large in size, very complex, lots of them, plentiful, and they look different than us, right? These are German cars. They're not American. They look very different than us. All right. So antibodies and receptors are specific to antigens. The antigen is the thing that doesn't belong. The antibody and the receptor are what are going to detect it in your body. So I started talking about this before. These little things are called haptens. Haptens are so small your body can't recognize them very easily. But when a hapten sticks to a cell, like a bacteria or anything, a splinter, anything that doesn't belong, now it's called an epitope. So it has a special name change. Haptens too small to recognize. Epitopes can be recognized, right? And if I look in really close at this epitope, it has a very unique shape to it. If I look at an antibody, that antibody has a unique cutout that fits perfectly around that unique shape. So an antibody is specific to one antigen. And then, like I said before, antibodies are usually drawn with Y's. So here you can see some of the cells we're talking about, B lymphocytes and T lymphocytes. So the B lymphocytes actually have antigens. Here you can see them sitting on the surface, and they're specific to a specific shape. Like this epitope will only fit that one, not these two. T cells have little receptors on their surface, and they do the same thing. They look for an antigen. So here's a little epitope on the surface, and they recognize it. This one's recognized, but that one won't recognize this. Right? The part that's binding, the part that's looking for the epitope, is called the paratope. So, and as an example, here's a paratope on the receptor right here in the very end. It's detecting the epitope. Right. So this kind of always puzzled me. If we only have 20 or 30,000 genes in our DNA, that's it. How is it possible that we can make literally trillions of different combination antibodies like this to recognize anything in the universe, basically, that is not you? These antibodies are designed so they can recognize anything in the universe, right? As long as it's not you. If it recognizes it as you, you don't want that antibody to be there because it could kill you. So I found out that it's a system called VDJ, system of recombination. So there are only a handful of genes actually that make antibodies. But what's interesting is there's another gene called the recombination activating gene that will scramble these genes and just take pieces of each of them. So let's say this code, like this for this combination, let's say it has a code from 0 to 9. That's 10 numbers, right? So this section goes from 0 to 9. This section goes from 0 to 9. So 10 numbers. This goes from 0 to 9, 10 numbers. Just by using 10 numbers, I could actually scramble these. I could go 001, 002, all the way up to 998, 999. There are actually 1,000 different combinations. There's 000 all the way up to 999 that you can make for this padlock. Your genes are the same way, except they're more complex because they don't stop at just 10. You can make literally trillions. And they estimate that you usually have about 10 million different antibody combinations floating through you at any time. So it's amazing how that happens. It's because of this recombination activating gene. It makes this random assortment of a padlock combination right here on this antibody that recognizes only one specific antigen. That's it. So the TCR and the BCR, this is T cell receptor and B cell receptor. They're all different. A B cell only has one receptor. It can only recognize one antigen. A T cell is exactly the same way. It has one receptor that recognizes one antigen. It can only attack that. So if you have two bad viruses going through your system, and this T cell is specific to one of those viruses, the other one could actually bump into the T cell and still just walk away from it. It's specific to one thing. All right, so we're going to expand on this a little bit. Two terms, clonal diversity and clonal selection. Clonal diversity is when your stem cells are developing. When they're developing into T and B cells, they go through that random assignment of a combination, and then they grow and develop. This is what's interesting. What if one of these cells randomly was assigned to look like you? If this cell looks like it detects you, it's killed. Because you don't want that thing getting out in your body. It'll attack your body. That would be an example of what type of a disorder? Where your body attacks itself. Autoimmune. Your immune system's attacking yourself. So any of these cells that are recognized as self cells are killed. So this one looks like, let's say that it was detecting heart tissue. Well, you don't want that thing to live because it'll attack your heart, so you kill it. If this one was recognizing myelin sheath in the brain, you don't want that to live because it'll kill your brain. You kill that cell. You don't take a chance at replicating. That's called clonal diversity. Lots of different combinations, but you're eliminating the bad ones. Right? This gives us something called self-tolerance. You're tolerant to your own tissues. Right? So anything that recognizes your own tissues and wants to attack it, that's killed. The next is clonal selection, and this is antigen dependent. Clonal selection, so these are floating through your body, even though you haven't experienced them in the past. But now let's say an antigen comes along. So let's say bee venom comes along. This is sensitive to bee venom, so it recognizes it. As soon as it's recognized, now this cell is going to replicate. It's going to proliferate. It goes from one cell, and then it turns into two, and it turns into 4, and turns into 8, and then 16, 32, and it just keeps going over and over and over again. So this is called clone selection. You select that one clone and you make lots of exact copies of it. Since this is sensitive to only one antigen, all of these are sensitive to the exact same antigen. So some of these will actually go out, they'll fight the invader, they'll kill the invader, and they'll die. Some of these will hide in the closet as a memory cell and come back later, so that when this invader comes back in the system, you already have this army of memory cells that are waiting for it. Right? So that's the difference. Clonal diversity is antigen independent. This is from stem cells. You randomly assign a combination. Clonal selection is antigen dependent. Now, once that cell has recognized the antigen, it can replicate and make tons of clones, and clones are all exactly the same. Hate to make a Star Wars reference, but like when they did the whole Clone Wars thing, they took that one soldier, that one bounty hunter, and they made millions of copies of him that were exactly the same. Every soldier was exactly a copy of that person. So next, what are professional antigen presenting cells? And this is kind of cool. There are cells in your body that will attack an invader. They take a piece of the invader and then they present it to the other cells. I like to think of them as headhunters, right? So what they do is, let's say this cell, this is a dendritic cell. It sits in your connective tissue under your skin. Let's say that, well, we'll take this pathogen. This pathogen comes along, it activates one of those toll-like receptors that are sensitive to PAMPs on the pathogen. It activates the toll-like receptor. Some genetic changes go on inside this dendritic cell. The dendritic cell will actually become mature. It knows what the invader looks like. It'll go up into the lymph system, and it will actually chew up, digest this pathogen, and take its head, just one piece of it, like the head, put it on a little stick, and parade that stick around the lymph node. It's kind of cool. It's like a headhunter that finds a victim, eats the victim, but keeps the head on a stick, and then parades around town and says, hey, look what I killed. You should get you some of that. These things are delicious. So now you've got this cell that's parading around the lymph node saying, hey, look what the invader looks like. It's delicious. You should go eat one. So now these T cells, they know what's going on, and they're going to go out there looking. They're going to try and find that invader. They know what the face looks like, and they're going to look for another invader with the same face to try and eat it and kill it. These are called antigen presenting cells. The two major ones, APCs, are dendritic cells and macrophages, which we keep coming back to this whole macrophage over and over and over again. So a macrophage will eat its invader, it'll chew it up, digest it, take what it wants and keeps it, spit out or defecate the rest out of its body through exocytosis, but it keeps one little fragment, just that little antigen, puts it up at the surface of its cell, and tells all the other cells, this is what the invader looks like, let's go get him. Right? So it's kind of cool, and I'll add these different steps to the process later, but just this communication. Dendritic cell detects that invader, it tells the T cell tells the B cell makes antibodies and just sprays the environment with these antibodies to try and attack the invader. So, and you can see the antibodies will actually stick to mast cells. So if the invader gets close to a mast cell, it knows what the invader is, and it will degranulate with what chemical? Histamine, right? So histamine, heparin, the chemotactic factors, but definitely want to remember that histamine. Okay, so describe the steps of antigen binding, processing, presenting. And this is kind of taking the antigen presenting cell and going a little deeper. So this antigen presenting cell recognizes the epitope. It pulls it in. So if this is a macrophage here, it swallows it up and pulls it in. It's going to present that piece of antigen on the stake. The stake or the stick or post or whatever you want to think of it is called an MHC, Major Histocompatibility Complex. It's on the next slide. But the name of the stick is an MHC. All of the cells in your body have MHCs on them except red blood cells. So all of the cells in the body have these little stakes. These stakes are special because the antigen's stuck at the end of it. So now this T cell comes along and says, hey buddy, how's it going? Recognizes the antigen and says, hey, that doesn't belong here. Where'd you get that? The macrophage has to squeal and say, hey, yeah, I was down on the corner and I saw it there. I ate it. It was delicious. Go get you one. They're down on the corner. So now this T cell's on the prowl. The T cell's hunting. It's hunting for wherever that antigen thing is. It's going to find a B cell, and it's going to tell the B cell, hey, that's what the invader looks like. Make antibodies. Let's get those antibodies out there, and they'll stick to the invader and slow it down so we can go ahead and eat it. So the antibodies are made and stick all over that invader so that you can eat it. Right? So some of these antibodies will float through and they'll stick to a mast cell. Not activated, they just stick there. But if the invader comes along and binds the antibody, then the mast cell degranulates. And I already talked about this, so releases histamine, heparin. Okay. What about a macrophage? If a macrophage recognizes this invader stuck to an antibody, what do you think the macrophage would do to it? Yeah, it'd eat it. Its job is a macrophage. It's a big eater. So it's out there trying to find things to eat. MHC is an important concept. So major histocompatibility complex, it's glycoproteins, little sugar proteins that are actually stuck on the surface of every cell in your body except red blood cells. So the genetic code are called HLAs. And we'll talk about this on the next slide. But the MHC is the protein, the HLAs are the genotype. So you've got a phenotype and a genotype. Right? Here's how it works. You find the antigen, you bring it into the cell, if this is a dendritic cell, you break up the antigen, you keep a little piece of it, and you push it along to the rough endoplasmic reticulum. Well, this major histocompatibility complex, the steak is in there. You just stick the antigen on the steak, pack it in a vesicle, ship it to the Golgi. Golgi puts a shipping label on it and puts it on the surface of your cell. Now I've got that steak on the surface of my cell with the antigen on the surface. It's such a cool design. This is going on in your body for thousands of years. Well, not your body specifically, but you know what I mean. And nobody had to teach it to do that. This is just something that just happens to work perfectly. So let's say here, this cell is infected by a virus. So the virus gets in, releases its DNA. Luckily, this cell recognizes, hey, you don't belong. It takes the antigen from the virus and says, this is what the virus looks like. It'll go through the same process here. It'll pop that virus antigen on the surface, but it would also make a cytokine chemical to release into the environment to tell its neighbors, hey, there's a virus present. I've got this one captured. I'm going to share what it looks like with the other cells, and then I'm going to kill myself. What's the name of that cytokine? That interferes with viral replication. It's an interferon. So haplotypes. Remember, haplotypes are the genome. MHC is the phenome, so phenotype. All right, here's how it works. So who's this? Is this mom or dad? It's round, pretty and shapely. It's mom. And then dad's the blockhead, right? So the code for these things, the little stakes, you've got this genetic code with lots of possible combinations. It's like a super complex padlock. Look at that. There's 0303, 08, 04, lots of weird combinations. Mom has two sets of these randomly assorted combinations. Dad has two sets of them. They don't look the same. Right? It's rare to actually accidentally find somebody that has exact same numbers here or the same combinations. So they have babies. And remember, when you have a baby will get one of these from mom, one of these from dad. So let's say that this first baby got this one from dad and this one from mom. Second baby got this one from mom, that one from dad. Third baby comes along, third baby got that one from dad, that one from mom, which looks exactly like the first one. So these two siblings have identical haplotypes. They have the identical genes for these MHC receptors. These two people would be ideal transplant recipients. So if this guy had a kidney that was broken, their sibling would be a perfect match. The MHCs on the surface of that kidney look exactly like theirs, so they can transplant, and it looks the same. Produces risk. Right? How about these two? How much are they compatible? They're only 50% compatible. This part and that part, which isn't close enough. So these two are not so compatible. This person, if they've got a kidney, if these two swapped a kidney, they'd be on immunosuppressants for a long time just so that their body wouldn't attack that kidney. Right? So think about it. Who would be your ideal person to get a transplant from if you could be any type of person out there? I'd be an identical twin. And I'd make sure that my identical twin was a very generous person. Because the identical twin, their haplotype's going to be exactly identical. So think about it. What are the odds of two kids in this family having the same genes? Not identical twins, but having the same genes. Well, you can do one pair would be this and that. Another pair would be this and that. Another pair would be this one, that one. Another pair would be this one with that one, and now we're out. So there are only 4 possible combinations. That means there's a 25% chance that one of your siblings will actually have the same haplotype as you. So it's kind of crazy. But every kid is going to have a 50% match with their parent. So nobody is going to have a perfect match with their parent because it's just not going to happen. That's like impossible. So the next thing you want to know about these MHCs are where they hang out. We've already talked about the process of making them. Antigen to endoplasmic reticulum, goes over the Golgi packages, ships them to the surface of the cell. So they're hanging out here on the surface of the cell. MHC1 are on all the cells of the body. MHC1 express endogenous products. So MHC1 will show your genetic code. It says, hey, I'm not only a human, but I'm Shannon's cell. So endogenous products. If a cell in here has a virus, it will say, I'm infected. Kill me, and puts a little label up here. Okay. That one that's infected with the virus may also show an antigen for the virus too. But the MHC1 is just endogenous products. It's telling everybody out there, hey, I'm specifically this cell. Right? Second are MHC2s. MHC2s are on B cells and antigen-presenting cells. So macrophage, dendrite, and B cells. These specifically put antigens on the surface. They'll take the antigen in, they'll actually put the antigen on the MHC and post it up at the front. Right? And we're not quite here yet, but I circled something that's kind of important. So when we talk about T cells down the road, you want to remember that these different T cells, if it's a CD8 T cell, it binds to MHC. So the moral of the story here is that CD8 T cells are what we call cytotoxic killer cells. Right? Those will bind to anything that's making endogenous products. It'll find a cell that has a virus in it and it's saying, kill me, and puts that little MHC1 on the surface. Cytotoxic T cells will kill it for it. Turn on apoptosis. Right? The second one's MHC2s are recognized by CD4 or the T helper cells, TH cells. So the TH cells are looking for the antigen presenting cells. These T helper cells are so important. They're like the general, the commander-in-chief of all of your immune cells, your adaptive immunity. All of the other ones look up to this CD4 to know what to do. So these are the scouts. They found the invader. They bring the antigen back to the T helper cell, and the T helper cell says, well, I know what these are because your MHC2 has the antigen. And then these T helper cells will share with everybody else. It'll share with other macrophage. It'll share with other T cells, other B cells. It spreads the word really quick. So homework number 7. Speaking of HLA matching, you're going to go to a bone donor site. So here's the actual address. You can type it in. You can Google it if you want. And then you're going to discover in there a couple things. I looked around the site and found these, so they're not always going to be extremely clear, but I want you to learn a little bit about bone marrow donations, and HLAs. So the first question is, between what age do donors have the greatest chance for a transplant? Not the recipient, the donor. So we usually think about the recipient being too old maybe to get a transplant, but what about the person donating the organ? Next, how likely will you match a patient? And if you go to this website, this is actually a fact sheet. This answers directly in that website. Okay. Next, what's the difference between bone marrow and PBSC? So I'm going to have you write down what it is. Tell me what PBSC is and what's the difference between that and bone marrow. Okay. And then also on that page, are there any risks to donating bone marrow? There are some risks. What are they? Okay. And then after you're done with that, I want you to think about it for a second. I actually told you the answer, but did you remember it? So you figure out what are the chances of being a match to a full sibling, a parent, and an identical twin. So this full sibling means that you have the same two parents. So you and your brother have the same mom and dad, not like half siblings where you share one parent. But for a full sibling, two people with the same parents. What are your chances of having a perfect match for a parent? And then what are your chances of having a perfect match for an identical twin? Go ahead and pause, write down, and I'll see you in a few.